Hi, I'm Dr. Shakti, and we'll be discussing about plasma membrane from an entrance perspective. Now, I uploaded another video where I put up a lot of questions, MCQ based questions as a test. So, in case you want to go do that first, I put a link over here. You can check it out. In case you've already done that and come here only for the answers, you can skip right to the end of the video where all the answers are given. And before that, we'll just go through the explanations to cover whatever is essential for understanding this topic from an entrance perspective. Okay. So in case you're wondering how to study this, these are basically the headings under which most of the questions come. So these are the concepts you have to know to answer the questions, most probable questions that are going to come out of this. Okay, so one is the percentage composition. Okay, then is what is phospholipid, what are the different types of phospholipid. Then fluidity and permeability. Based on this, a lot of questions are coming. Okay, now role of each part, like what is the role of protein, the lipid, the carbohydrate layer. So that is not something that you need to know in detail for MCQs. Okay, so I'll be giving a more detailed explanation on it in another video. Okay, but for an entrance, I don't think you need to break your head over trying to understand it in detail. Okay, so in case you want to learn it, I'll put another video up. Okay, so you can learn it in that. So first set of question is gonna be like this. One is main composition of plasma membrane. Different substances are given. Eukaryotic plasma membrane is made of all except. So by this, basically you need to understand the different composition of the plasma membrane. So if you take a look at the plasma membrane, there is protein, there's carbohydrates, there's lipid. Okay, this protein structure that accounts for 55%. So majority, almost majority is proteins. Okay, so if a question comes, which is a major component in the plasma membrane, that is protein. Okay, next is your lipid. So in lipid, your phospholipids are majorly present. So that is around 25%. Then comes your cholesterol, that is 13%. Then there are other things. Okay, so like sphingomyelin and stuff like that. So all that accounts to 4%. Carbohydrate accounts to very little, that is 3 to 4%. Now you can see over here, these are the phospholipids. You can see the carbohydrate moieties on top outside. There are protein structures over here. So this protein, this is the integral protein. And there are peripheral proteins. Mind you, peripheral proteins can be inside as well as outside. Okay, so they are peripheral, not going in through the phospholipid layer. Okay, so there's integral protein, peripheral proteins, there are phospholipids, there are carbohydrate moieties like this. And this is the percentage that we need to deal with. So next question is on weight basis, the membrane contains protein and lipid in ratio, decreased protein ratio, highest protein content. So these set of questions deal with the protein to lipid ratio. So this is a diagram from Harper's textbook, okay, biochemistry. So what you can see is the mass ratio of protein to lipid. Now, a normal cell, most of the studies were done in human erythrocyte because it doesn't have a nucleus. It was easier to study it. So the structure of the membrane. So you can take that as a reference. That is 1.1. So almost, 1:1, right? Even 55% and 42% that we had told earlier. So that almost comes to 1.1. A little more proteins extra. That's it. So you can say if options come 1:1 would be an ideal answer. Next, you can see outer membrane of the mitochondria. So the mitochondria has an inner membrane as well as an outer membrane. So the outer membrane again is similar. Sarcoplasmic reticulum is a little more high, more protein is there. So that is 2. Okay. And inner mitochondrial membrane is the maximum one, where protein is maximum. Okay, so compared to outer, inner membrane is 3.2. Okay, so this is a ratio. And the least is for myelin. So question normal is 1:1. Maximum protein content is for mitochondrial inner membrane. Minimum is for myelin. Next set of question, the phospholipid that is present maximally. So different types of phospholipid are there, and components of cell wall with carbohydrate moiety, and different substances are there. So for this you have to know a few details about what are the phospholipid and different types of phospholipids. So the base structure of a phospholipid, as you can see over here, it has a glycerol part over here. CH2, CH, CH2, OH, OH. So that glycerol, to that there is fatty acid residue. Okay, acyl chains are there like this. And on the other side, you have a phosphate. So we have a glycerol, the fatty acid chain, the phosphate. Okay, and to that, on top of it, something else is added, and that determines what type it is. For example, this is glycerol based, and if ethanolamine is added over here, it's going to be phosphatidyl ethanolamine. Instead of this, if serine was added, or choline was added, or inositol was added, accordingly it would have been different substances: phosphatidyl, serine phosphatidyl, choline phosphatidyl, inositol. Okay, the base structure is the same. It is glycerol based. So these substances, all these are what are called phosphoglyceride. Okay, so out of the phospholipids in the plasma membrane, phosphoglyceride are the major component. So apart from phosphoglyceride, what else are there? So I told you this is glyceride based. Okay, so now instead of this glycerol, if we replace it with something like sphingosine, you can see the structure of sphingosine here. Okay, everything else is same: phosphate, fatty acid chain. Okay, and here you can see choline. So this is what is a sphingomyelin. Glycerol replaced with sphingosine. You got phosphate chain, fatty acid chain. Here there's a choline. So this is sphingomyelin. Similarly, sphingosine, fatty acid. Instead of a phosphate, you would actually add a galactose over there. So this is the galactocerebroside. And just like this, there are other structures like the ganglioside. Okay, so all these are sphingosine based. Okay, so now that is done, we'll check the next question that is there. So this talks about lipid bilayer in general. They've been asking about asymmetric arrangement, symmetrical arrangement, lateral diffusion of protein and lipid. So all these answers are based on this concept. We'll just understand what it means. So when you take a look at the bilayer, you have the phospholipids over here, the hydrophilic part. This is the water. So water is there in the extracellular fluid, water is there in the cytosol. Okay, inside. So ICF, ECF, that is where the hydrophilic parts of it are going to be there. And in between them is the hydrophobic part, where the fatty acid chain is there. Now, the lipid composition, the phospholipid composition in the two leaflets. So these are called the leaflets. So in the two leaflets, they are not same. What is seen in that outside, most likely is going to be a phosphatidyl choline. On the other hand, inner side, the cytosolic side, it's most likely going to be something of phosphatidyl inositol, ethanolamine, or serine. So there is functional differences. For example, for protein kinase C activation. Okay, phospholipase C, then finally protein kinase C, so IP3, DAG, and all that to happen, this part should be broken down. So this is there functionally, it is there inside. Signal transduction and a lot of other things. Okay, so that is one reason why such a asymmetry is there. So that is the first thing. There is a asymmetry between the two leaflets when you talk about lipids. Now, this is hydrophobic. So obviously, things that dissolve in water and have to go in through channels and pores, they cannot move through this laterally like this inside the membrane. Okay, if there's a pore, then it goes through the pore and goes inside. Otherwise, it's not going to cross this. It's not going to move laterally and all through this. On the other hand, it's seen that lipid and protein structures have a lateral sort of movement, especially when it comes to renewing the cell membrane after damage. You can see that these keep, moving laterally. So that is another thing. Next question is going to be about the fluidity and the role of cholesterol. Okay, so different types of acids are given. Yeah, fatty acids. We'll come to understand what that means. So this again is your phospholipid. Okay, this is the hydrophilic, hydrophobic part. Okay, now if you can see over here in this fatty acid residue, there is a double bond over here. Okay, so that leads to some sort of a kinking. The shape, you can see it over here, slightly bent to one side. So this is your unsaturated fatty acid, this one. Okay, and over here you can see no double bonds, no kinking, saturated fatty acids. So when questions come, you have to understand few things about what increases fluidity. Okay, this again I'm not going to go too much into detail, because for an entrance perspective you don't need to go into too much detail. You just have to understand few basic things. Okay, later in other videos I'm gonna upload more concepts about this. So first thing for fluidity, high temperature. So if the temperature is high, then fluidity increases. Double bonds, more the number of double bonds, more unsaturated it is, more the fluidity. Okay, and if the fatty acid chain is shorter, that again. And if the concentration of cholesterol is high, that also increases fluidity. So things that decrease fluidity will be the opposite of this. Okay, so questions come based on this, you just remember these points. So if you want to remember this, since questions come and these are the options that have to be there in case you don't know the biochemistry of that, very basic idea of it. You can see over here on this side, this is the number of carbon atoms that are there in the backbone of it. Okay, and this is the number of double bond. So in this fatty acid structure, stearic acid has 18 carbon atoms backbone, and it has no double bonds. This is what it means. So because it has no double bonds, it's going to be saturated fatty acid. Okay, as you keep going down, you can see that the number of double bonds are increasing. Okay, now oleic acid, linoleic acid, alpha linolenic acid, all of them have 18 carbon atom chain, but the number of double bonds keep increasing. Okay, and eicosapentaenoic, docosahexaenoic are not 18 anymore, instead they're 20, but they have 5 and 6 double bonds. So as you keep going down, then the fluidity actually keeps increasing. So if two of these are there, then the one with the greater number of double bond is going to be more fluid. Next question, most permeable to pure phospholipid bilayer and most diffusible ion across membrane. So again, here the concept is the difference in permeability of substance. So this is a diagram from Harper's textbook from biochemistry. So basic thing is fat soluble substances are most easily diffusible. Okay, because they have to pass through the hydrophobic phospholipid layer, and it has to be fat soluble for it to pass through easily. Okay, so that is the ultimate option. If these things are there, this is going to be the most easily passable one. Okay, the permeability for these are going to be the best. Okay, in case these are not there and these are the options, then you'll have to think. Okay, so for these are basically water soluble. So if there's a channel, a carrier, or any sort of pore, pump, whatever. Okay, but for them to move, water has to be there. They are water soluble substances. They go through these channels. Okay, now water has the highest permeability over here. Now usually questions come among these ions. Out of these ions, what you can see is chloride has a maximum permeability. Next is potassium, least is for sodium. Also, you can see glucose, urea, where they stand. So you just have to remember these basic things. So water, indole, urea, glycerol, tryptophan, glucose, chloride, potassium, sodium. So this is the order of the permeability. And of course, fat soluble ones are the highest. Lipid raft. So for that you need to understand what a lipid raft is. Okay, so since it's been asked, I'll just discuss what it is. So it is a particular area. You can see that there are certain microdomains which are rich in cholesterol, sphingomyelin, glycosphingolipid. So you already know what cholesterol, sphingomyelin and all is. Okay, so these are rich in these substances. Okay, so there are certain microdomains which are rich in this. Okay, and they seem to be specialized and are thought to be playing a role in signal transduction. Okay, so for signal transduction, this is important, these lipid drafts. Okay, lipid draft also comes into play when you talk about, caveolin. Okay, so all that transport through the membrane. Next is about RBCs. That also another thing that I've seen. Few questions about RBCs membrane. So what you have to understand is the structure. So this is the inner part. Okay, this is the inner part. You can see over here a glycophorin, then ankyrin, spectrin, band 3 different substances. Band 3 is an integral protein which passes through transmembrane integral protein. Okay, and this is basically a ion transporter, a bicarbonate chloride exchanger, that is nothing but a anion exchanger. Then we have spectrin and ankyrin. Okay, so this is a structure of spectrin. With anchoring, it is anchored into other integral proteins. So these are peripheral proteins. These peripheral proteins help by binding to actin. So that helps maintain the shape and membrane integrity and all that. So these are important for those in RBC. And there are substances like glycophorin. So glycophorin has a lot of carbohydrate moieties in it and sialic acid residues in it. So that kind of gives a negative charge to the RBC membrane. So these are all substances that are there in RBC. So we'll come to those questions and try to understand the answers. Yeah, the main composition of plasma membrane is, so the answer that is protein, 55%. Eukaryotic plasma membrane is made of all except, so carbohydrate yes, cholesterol yes, lecithin, triglyceride no. So phospholipids are there. You see that there are two fatty acid chain. In triglyceride, what has happened? One glycerol chain is there with 1, 2, 3 fatty acid residues. So that is not what is there. Okay, so this is what is not there in the plasma membrane. On weight basis, the membrane contains protein lipid in the ratio. So normally it's going to be 1:1, almost. Yeah, there's no other better option. Okay, decreased protein ratio is seen in. The protein ratio decreased as in the protein is going to be lesser. So usually in myelin sheet, the ratio is less. And the highest protein will be your mitochondria. Which mitochondria, outer or inner? So it's going to be for inner. Phospholipid that is present maximally. So we had seen this. Phosphoglyceride is the answer for this. So that includes your phosphatidylethanolamine, serine, choline. Okay, so that is your major component. Next, components involved with carbohydrate moiety. So phosphoglyceride, triacylglyceride, cholesterol, no. Sphingomyelin, no. That is choline and all. So out of the option, ganglioside is there. So ganglioside and galactocerebroside. So these are things that have some carbohydrate element in it. Okay. So here the answer is going to be ganglioside. False about lipid bilayer. Is it asymmetric? Yes, there is asymmetry. Lateral diffusion of protein and lipid and not ions. Made up of amphipathic? Yes, phospholipids are amphipathic. Okay, one terminal is hydrophilic, other is hydrophobic. Symmetrical arrangement of cell membrane component, that is a false statement because it's not symmetric, it's asymmetric. In cell membrane, true statement is. So, lipids are arranged symmetrically, proteins displayed laterally, lipids are regularly arranged. So again, it's the same concept. So here, the thing is proteins get displayed laterally. So these are just questions that have been asked previously, which I've seen in many question banks, so I just put it up. Membrane fluidity is increased by. So this is not gonna be there because it's saturated. Linoleic acid is there, so that's gonna be a option. Function of cholesterol in plasma membrane. Cholesterol helps to maintain fluidity. Okay, and especially in high concentration, it increases the fluidity, and much lower concentration, it kind of decreases the fluidity. Most permeable to pure phospholipid. So it's a pure phospholipid layer. So there are no proteins, channels, anything. So what is going to pass through? Something that is hydrophobic. Okay, lipid soluble. So that is going to be oxygen. So if this is the option, that is the answer. most diffusible ion across the membrane. So out of all this, chloride will be the best bet. Lipid rafts are seen in your plasma membrane. Okay. Membrane integrity of RBC is due to. So here the answer is going to be spectrin. So in case you have a confusion between spectrin and ankyrin, go for spectrin, because spectrin is the important component over here. Ankyrin is something that anchors your spectrin into a integral protein. Okay, but the most important component over here is spectrin. Glycophorin is present in, so this is again RBC. Okay, so I hope this was helpful to you. In case you have any doubt, feel free to reach out to me. Okay, so I'll be putting up more content like this. So anyway, thank you.